We continue with the second video, which is of transport and plants, O-level biology 5090 syllabus, and we're going to study the parts of the chapter which was left, which is the rate of transpiration. So if we have to investigate, so you have to give me practical procedures how we're going to investigate the rate of transpiration and what are the factors that affect the rate of transpiration. It's very easy to remember. I've made a mnemonic HALT. HALT, so humidity, air movement, or you can say wind, and light. What does happen to the rate of transpiration in light? So you'd have to compare it with keeping the plant in the dark. And the effect of temperature. If we increase or decrease the temperature, does it have an effect on the rate of transpiration? Yes, it would, because increasing the temperature would increase the rate of evaporation. So more evaporation, more water vapors in the air spaces. More waters in the air spaces, more water vapor will diffuse out. Now the instrument that we use in studying the rate of transpiration is called a potometer. There are many different types of potometers, and we will discuss these different types and see how we measure the rate of transpiration. Now if you look at a potometer, it's got basically a few things which are common to all potometers. Number one, there has to be a part where you put a cut chute and you seal this. Why you seal this? Because you don't want water evaporating from here, because then you would say this is not measuring the rate of transpiration. So the rate of transpiration means we want to somehow measure how much water is coming out of the leaves. So you have another water reservoir here, and you have a tap here. This is the important thing, the tap and the water reservoir. And then you have a capillary tube. Capillary tube, why? Because it's a very narrow tube, so even if a little water movement takes place, you can register it and you can see it, you can measure it. And you have a continuous, now all this is a very continuous water here, this water here, this here, the water here, which is all in blue, is all the water, and there should be no air bubbles in this. So this is all the water, and then this capillary tube dips into this water, and you have introduced an air bubble. Now what is going to happen is that the water is going to move in. I'm just going to use another color to show that. So the water is going to move in, and then it's going to go into the plant, and then it's going to come out of the leaves. Now this bubble is going to move this side. Why? Because this water which was inside this tube is going to come here, and then it's going to move here, and going to move into the plant, and going to come out of the leaves. So the air bubble is going to move this side towards the plant, and the length that it covers, the length that it moves, is going to be measured. So say it measured 10 millimeter. So the bubble moved from here, the bubble moved 10 millimeter to this point. So that means this fluid, which is 10 millimeter in length, so it's a column. And you know how do we calculate the volume of a cylinder? It's pi r square h. So this length of the column would be h, and then if we knew the radius, if you know the diameter, then you can calculate the radius, so you can calculate the volume. But another way that you can calculate the volume is that you open this reservoir, and if this reservoir is calibrated, then you would know how much water would it need to send the air bubble back to its initial position or the zero position. So a very basic method is that we're going to measure the length of the column. So 10 millimeters is the length of the column in which the air bubble moved. Finding out the volume is of course a separate story, and how we do it, I've just told you one method and I'll give you another method. So the air bubble, the movement of the air bubble is telling you how much water moved into the plant and out of the plant. So a potometer, we say, measures the rate of transpiration. Now I disagree. It does not measure the rate of transpiration. And I usually give you time to think about it. It is not measuring the rate of transpiration, because what you are assuming is, you are assuming that all this fluid, whatever water was here, has gone in, and all of that water has moved out of the plant. Well, that's not true, because some of the water has been used. Some of the water has been used by the plant for hydrolysis, for photosynthesis. So you are thinking that all the water absorbed has been transpired, has been diffusing out of the leaves, has been moving out of the leaves, or has been diffusing out of the leaves. So what we have to understand is that there's a very small volume which is used by the plant. So that's why we are assuming that whatever water is absorbed is actually lost from the leaves as well. So this is an assumption that we make, that the water absorbed is the water transpired. So, but actually this is only measuring the rate of water absorption by the plant, not the rate of transpiration. So we do ask you that question at times in the exam, that what is this potometer measuring? And you say it's measuring the rate of transpiration. And I say, no, I don't agree. The reason why I don't agree is because it's only telling you how much water went into the plant, but it's not telling you how much water left the leaves. So please understand, this point of mine is that we are not considering the fact that water might have been used inside the plant, in the cells of the plant. First, let's look at the effect of humidity on the rate of transpiration. And I've mentioned this many times, rate is something to do with time. Rate is always something oriented with time. So rate of transpiration. So when we're going to study humidity, well, we're going to study the potometer without any, we're going to keep lamp here. We're going to keep the temperature constant. So this is going to be in a room which the temperature is maintained. So temperature must not change. We've got a lamp here, so that lamp is at a certain distance from the plant. And so light, temperature, there's no fan on, it's just still air. And we want to study the effect of humidity. So what we're going to do is, we're going to place a plastic bag around this. So in one experiment we have the plastic bag, in another experiment we don't have the plastic bag. Now when we have the plastic bag, what is going to happen is that the water vapors which are going to diffuse out are going to condense in the plastic bag, and that's going to increase the humidity. So when we're going to study it, we're going to study that did this plastic bag make any difference to the rate of transpiration? Did it sort of move, the bubble moved faster or the bubble moved slower? Naturally, if you increase the humidity outside, then the rate of transpiration will decrease, because if you remember, I told you earlier, this is a leaf. And if there is too much humidity outside, say there is 100% water vapors outside and there is 50% inside, well, then the water vapors are not going to diffuse out, and outside is 100%. So too much water vapors outside will in fact stop the rate of transpiration, because these water vapors now cannot move out. They will stay in, they will not be able to diffuse out, because there is no gradient, because outside there is a lot of water vapors outside the leaf. So increasing humidity will decrease the rate of transpiration, and the opposite. If the humidity is less outside, naturally water vapors will diffuse out. So this is the first, the mnemonic what I gave you was HALT. So we said we studied humidity. Now in the next one we're going to study air movement, and that is having some sort of a fan which is going to produce air movement or wind. Now the second factor that we're going to study is air movement or wind. So you're going to place a lamp. The lamp is going to be at a fixed distance, but what you're going to now place is a fan. So I'm sorry, I can't make a very nice fan, but the fan is going to be at a certain distance from the plant. And so you'll have to do an experiment with the fan and one without the fan. So one fan present and one no fan. And then you note the movement of the bubble. Naturally, if you have the fan on, what is going to happen? This is the leaf. If you have the wind blowing, then the water vapors which have diffused out are going to be carried away. So the air movement is going to carry away the water vapors. So always there will be less water vapors outside the leaf. There will always be less water vapors outside the leaf. Because what is happening is that when you have the fan working, the air is moving and the air is carrying away the water vapors around the leaf. So always there is a diffusion gradient. Inside the leaf, inside the leaf there is more water vapor. And outside the leaf there is less water vapor. So water is going to diffuse out. So water vapors are going to diffuse out. So if we increase the wind or we increase the air movement, then more water vapors would diffuse out of the leaf as compared to still air when there is no air movement. So this is what we have to understand about the second point, which was the air movement. Now the third factor that we need to study is the effect of light on the rate of transpiration. Now you place a lamp and then we study the rate of transpiration. So one will be with the lamp and then no lamp. So one in light and one in darkness. Now when we study the rate of transpiration, it is naturally the light, the rate of transpiration is more because light results in the stomata opening. In dark, the stomata close. So in light, the stomata open. There's a cue to them to open because light encourages them to photosynthesize and the carbon dioxide is going to diffuse into the leaf. So light will result in naturally more photosynthesis and more transpiration because the stomata are open, so water vapors can diffuse out. So the effect of light would be more transpiration, effect of dark would be less transpiration. There's some of it, of course, does occur because of cuticular transpiration. Then we study the effect of temperature on the rate of transpiration, and for this you'd have to place this in a water bath. You'd have to place this in a water bath, and this water bath would of course be maintained at different temperatures. So the water bath would be maintained at different temperatures depending on what temperatures you want to take. So 30 degrees Celsius, and then you measure the movement of the air bubble, and then you measure it at 35 degrees Celsius, and then at 40 degrees Celsius, then at 45 degrees Celsius, and then at 50 degrees Celsius. I've always said you use 5 temperatures. So of course, increasing the temperature would increase the rate of transpiration. Why? Because increasing temperature provides more kinetic energy. So more kinetic energy means more rate of evaporation and more rate of diffusion. So two things will increase. Number one, the rate of evaporation will increase, and the rate of diffusion will increase. So as we say, increasing temperatures will increase the kinetic energy. So more evaporation and more diffusion. So when there's going to be more evaporation and more diffusion, so it's going to result in more transpiration. So more water vapors diffusing out. But then of course, humidity would be again a feature which would come into play. Because if there is more humidity outside than what is inside the leaf, then of course even the evaporation and diffusion is not going to help. So these are the four factors that we needed to study: the effect of humidity, air movement, light, and temperature on the rate of transpiration. Now the next syllabus point which we need to discuss is investigate using a suitable stem the pathway of water in a cut stem. Now this is what you don't read. The pathway of water in a cut stem. So something you've got to do is, number one, you've got to first prove to me that the water molecules from the soil have gone all the way up and into the leaves, And then I want you to tell me where did they travel in the stem. So I need to cut a section of the stem. And if you remember, the section of the stem was like this, and the vascular bundles were on the sides. Now there are two biologists that are arguing. One biologist say that the water molecules travel in the xylem, and the other biologists say the water molecules travel in the phloem. Now one is wrong and one is right. Now how are we going to prove this? Well, we all know, yes, that the water travels in the xylem. But how are we going to prove this? So what do? We do an experiment. We do an experiment is that we place this cut, we take a cut stem, we don't take the whole plant with the roots. We just take one branch, say this branch, and this branch has got, say, 4 or 5 leaves on it. So we cut this branch here and we use this cut stem. So it says cut stem. Please understand this. We say cut stem. So this cut stem has to be used, and we put this in a beaker. So we take a beaker and we say put some water in it and we put a dye in it. And we put a dye in it, say we put 1 ml of methylene blue. And then we take a cut stem, it's got these leaves on it, and it's got a white flower. There's a reason why we take a white flower. Because I want to find out when this color has gone in and it has reached in the flower as well, and the flower is now blue in color. Because it was a white flower, that's why I could see this color change. If it wasn't a white flower, I would not have seen this color change. Now what I do is I take a section of the stem here, a very thin section. I take a glass slide and I place this thin section of the stem on it. And then I look at it under the microscope. When I look at it under the microscope, I find, you remember the vascular bundles were on the sides. And now where do I find the blue dye? The blue dye is on the inside. Now this factor that the blue dye is on the inside, now if you remember earlier in the chapter I told you that the xylem is on the inside. So the two biologists arguing about the fact that does the water and ions travel in the xylem or in the phloem, well then they would then come to an agreement that yes, because this dye, this blue dye, was on the inside of the vascular bundle. But if it had been on the outside of the vascular bundle, then of course we would have said, okay, well, they travel in the phloem. So now if you look at this point, it says investigate using a suitable stem the pathway of water in a cut stem. So we wanted you to tell us whether the water travels in the xylem or the phloem. So you then placed a stem with white flowers. You place this in bright light. Why in bright light? Because you see, in bright light the stomata open and the water moves out of the leaves. The water diffuses out of the leaves. The water vapor diffuses out of the leaves. So the water vapor diffuses out of the leaves and this creates a suction force. And the water is pulled up in the stem. And that is called the transpiration pull. That's another point of the syllabus which we need to talk about. Transpiration pull. So the fact that the water vapor is diffusing out creates a suction force. Just like I was telling you about the straw. When you want to suck the water up in a straw, well, you have to exert some force on it, some suction pressure on it, so that the water is sucked up the straw. So when the water moves out of the leaves, this creates a suction force, and then this water is pulled up in the stem. And this water is pulled up in the xylem. So that is the transpiration pull, which is because of the water vapors diffusing out of the leaves. The next point of the syllabus is how wilting occurs. Now, wilting occurs is when water absorbed excessive transpiration causes a plant to wilt. But basically, is when a plant loses more water through the leaves than it absorbs through the roots. So now that could be because there's less soil water. If there's less soil water, then the plant is going to wilt because transpiration is taking place. Still, the water loss is taking place from the leaves, and there is no soil water or there's less soil water. So the leaves droop. And when the leaves droop, stomata close. Also, plant hormone is released, which is called abscisic acid, and that results in the stomatal closure. Now, when the stomata close, in a way, it is saving the plant because less transpiration is occurring. But of course, photosynthesis is compromised. But if it rains, now we're thinking of a plant in a forest, when the plant will recover. So, this is in a way sort of saving the plant for a short period of time. But if, of course, the water loss keeps on continuing and there is no rain, then the plant will eventually die. So we have to know how wilting occurs. The next point of the syllabus is relate the structure and functions of root hairs to their surface area and to water and ion uptake. Water and ion uptake. So we have to discuss the structure and function of root hairs. So what is a root hair like? Well, simply we've done this many times. The root hair has got this long cytoplasmic extension. So a root hair has this large, the surface area is increased because of this huge cytoplasmic extension. Now you've seen these root hairs on carrots. If you've ever looked at carrots, what that is, the hair on top of it is which you remove, and when you should remove it because it's got a lot of mud sticking to it. Now those are increasing the surface area immensely. Normally plant cells are not like this. Normally plant cells are just like this. So this has this long extension, the cell wall and the cell membrane and the cytoplasm. So this makes the root hairs having a very large surface area. Now, how is it going to help? Now, first we've got to understand is that there would be the normal cell wall of a root hair, and then there would be the cell membrane inside it, which is very closely attached to it. So the cell membrane is there, and then in the cell membrane we have channel proteins, and the channel proteins are for the ions. So the ions are going to be transported by active transport, and they might be less on the outside, so 10 here and 20 inside, but still they would move in by active transport. So ions taken up by active transport, and of course the water molecules would enter by osmosis. Why? Because there's a higher water potential. There's a higher water potential in the soil, more water in the soil. And why is there a lower water potential inside the cell? Because there are other soluble molecules inside the cytoplasm and ions inside the cytoplasm and the vacuole. So water will move in by osmosis. So water from the soil will move in by osmosis, while the ions would move in by active transport. So the syllabus point is relate the structure and functions of the root hairs to their surface area and to water and ion uptake. Plus what you need to understand is that inside the root hair cell there would be large number of mitochondria. Why would there be large number of mitochondria? Because there's a lot of active transport taking place. So in any cell where there is more active transport means more energy needed. More energy means more ATP, which is the energy currency. So more energy needed. So that means because ions have to be taken up by active transport. So please understand the structure of a root here. How, number one, it's a large surface area because of this extension. And then the water and ion uptake will have proper protein channels in the cell membrane, which would help in the active transport. Now, the last as it is, this is I've completed the chapter. A quick recap. First, it said relate the structure and function of the root hair, which I just did in the end. Then it says state that transpiration is the evaporation of water at the surfaces of the mesophyll cells, followed by the loss of water vapor from the leaves through the stomata. Then the C part says describe how water vapor loss is related to cell surfaces, air spaces, and stomata, which I've discussed. Then the effects of air currents, variation temperature, humidity, light on the rate of transpiration. There's again experimental procedures, then how wilting occurs. Then investigate using a suitable stain the pathway of water in a cut stem. Then explain the movements of water to the stems in terms of transpiration pull, the suction force which is exerted by the movement of the water vapors or the diffusion of the water vapors out of the leaf. And then it says identify the positions of the xylem and phloem tissues as seen in the transverse sections of unthickened herbaceous dicot roots, stem and leaves. So we've got to see how do roots look like, how do stem look like, and how do the leaves look like, and when you look at a cross section of it, where is the xylem and the phloem present? And then of course the last is state the functions of the xylem and phloem. So please read through the book, and then you can watch the video and then again go through the book, and please do the past paper questions on this. And then thank you.